Hey, a woman gave me some soil at the LWA conference. I'm sorry, I can't remember your name, but you know who you are. So yeah, here's what I promised. Here's your soil, and this is from. Let me just get this bag out here. Something marked up on it. This is two plot five. So yeah, I noticed this little bit of aggregation, and this is where a lot of the action happens usually in the microscope. And yes, indeed, you've got little actinobacteria there on the west side of that aggregate there. So that's an aggregate. That's like a water stable aggregate you've got there. That's a whole load of organic matter and nutrients pulled together, clays, silts, sands, the whole lot all glued together with biological action. You can see some bacteria. You maybe haven't got your eye in yet, but if you look in the white space, all the bacteria are sort of moving around, hustling and bustling. But you can actually see some bacteria that have adhered themselves to the surface of that aggregate as well, just all around the sort of periphery of it. So that's pretty cool. But yeah, a lot of things. This is the food source for a lot of species. And obviously there's a lot of grazers that are attracted to aggregates, so a lot of predators come along. So if you have like a time-lapse camera or something, it's often a really good place to set up your scope and just sit it over an aggregate, and you'll get a lot of action. It's a bit akin to like an oasis, I suppose, in the sort of African Sahara. So but yeah, that stringy thing is a bit of actinobacteria, which is a colony of filamentous bacteria. They were recently called actinomycetes. And they're responsible for that geosmin kind of chemical smell, fresh soil smell after rain is all their sporing that you can basically smell. And yeah, I read in a paper recently that humans are particularly sensitive to smelling that. For what function, I'm not sure, because often actinobacteria are not sort of associated with in high populations, are not associated with particularly healthy soil. So maybe it's a warning mechanism to stay away from this patch of soil, or maybe don't eat the animals that are on this patch of soil, or something like that. And completely we're misinterpreting it these days. But yes, it's the mating ritual that you're essentially smelling, as horrible as that sounds, of actinobacteria. So they exist in vast numbers in soil, and they're kind of quite hard to spot in the microscope. Often you can see it's just a little tiny hair there, basically, if I'm not so zoomed in. And just going to go along and zoom in on any interesting structures that I find. Yeah, these look like insect parts, or possibly trichomes of plants. I'm going to say actually probably an insect leg or appendage of some kind, and probably not a trichome, just because of its kind of fuzzy nature. Trichomes are also fuzzy, but it looks to me like the entomologist in me is saying that that's an insect leg, so that's kind of interesting. You find them surprisingly often, actually. I'm not sure what they're doing, so we're just losing all the legs all the time, but nature moves in mysterious ways, I guess. Yeah, this is a maybe body of... No, it's not. It's a bit of plant material. So yeah, it is worth bearing in mind that, of course, a lot of things have probably gone dormant because they were sitting in those plastic bags for a wee while now. So I'm actually only getting around to doing the soil sampling today, which is the 25th, Friday of October. So I would imagine if I pulled this sample straight from your soil, we would see a lot more action probably. Also, it's the winter, so things are going into hibernation. No more exudates from the plants equals very little activity from the microbes because they're not being fed anymore. So those that have some sort of symbiosis or rely on plants for their source of carbon and energy often go entirely dormant. And this looks a little like an egg of a nematode. They sometimes, some species have the sort of pursed ends, just like a mermaid's purse that you find, for skate eggs. They can sometimes look like that. Sometimes they can look very cylindrical, like a giant bacillus bacteria. But anyway, there's nothing inside that, so unsure what that could be. This could very well be like some sort of spore. Could very well be a cyst of some amoeba. I think it's probably more likely to be a spore, actually, or a piece of pollen. So let's see what else we can find if I can like move the right direction into the sample rather than out of it. Obviously, as a grower, you'll be interested in the fungal populations here. So I'm on the lookout for hyphae. Yeah, that's just 2 bits of organic matter that have managed to attach themselves together. Let's just zoom all the way out, and then we can get better overview and more quickly find likely hyphal strands. Let's just zoom in and see if that's one. You're excited. Nope, that's a little bit of organic matter there. To me, like, sort of impressions, a bit hard to tell during the winter, so don't want to draw any false conclusions on this. Let me just try and tidy up this modeling a wee bit. There we go. I'm seeing quite a lot of sort of minerals kind of everywhere scattered about. So I interpret that as being quite a lot of soils where there's high mineralization basically, and it's there's a lot of immobilization, so there's a lot of minerals that are sitting and haven't been quite incorporated into the soil structure yet. So yeah, like often you see all these little shards of glass, and you can see little silt particles, clay particles are about the size of bacteria, so they're harder to distinguish. Let's just see what we got here. I think this is another bit of plant material of some kind. Yeah, it is some sort of plant root maybe. Looks like I could see a xylomorphlum there, so but yes, like I was saying to you when you drop them off with me, the fungal hyphae can sit around for quite a long time. They're kind of relatively recalcitrant. I mean, things will feed on the cytoplasm first, but especially some of these higher order fungi, the ones that you're really interested in, that establishes very strong mycorrhizal connections. They often, have a very recalcitrant cell wall. It can be from somewhere from, the C to N ratio can be like 100, or up to 1,000 sometimes. So, they tend to last in the soil, and they leave these little skeletons for you to find, like do a bit of a post-mortem on. So yeah, it'd be interesting to know if you could tell me in the chat what the treatment the soil's had, and if it's proving productive for you. So yeah, I haven't seen really any fungal hyphae at all yet. A few spores here and there. I don't think this is one, but quick look. Yes. I'm saying that because it looks a bit unusual, but just zoom in further and confirm that you are. Yes, indeed you are a fungi hyphae. So you can tell that straight away because it's got a cell wall, it's got a septa there. So that's one cell length that it's grown there, put down a septa. Got another septa there, a much lighter coloured one. So it's aseptate. But excellent colour. Can't see any oxalate crystals. It looks to have colonies of bacteria feeding on it. So, which is entirely unsurprising. The fungi will most likely be dead or dying and no longer able to properly defend itself from attackers. So that little gobbet that you see there, that's a load of fungi that are feeding on the juices as they're seeping out. And they might bore their way through the actual cell wall and through the cell membrane and into the cytoplasm, into the cell itself at the moment, and feeding on those nice nutrients. So yeah, but that's a good sign. So that is a classic fungal hyphae there. Mary in the chat will maybe tell you a bit more about what that could possibly be. As far as I'm aware, it's impossible to identify the species from, or most fungi, it's impossible to identify from species just on the hyphae alone. So, but yeah, I normally go for basidiomycetes if it's that kind of color. This looks to be another hyphae. Yeah, I can see cell walls there if I was very carefully zooming in out right in the center. So yeah, either recently germinated and it's not had a chance to put down its melanin yet. So it's very pale. or it's one of the quicker growing species that are more adapted to quicker, to rapidly changing conditions, and can jump off the back there and take advantage of those where other fungi can't. So yeah, something that requires a bit less stability in order to occupy its niche. So yeah, I mean, you'll see if you watch my other videos, but I usually say seeing about a fungal strand at this kind of magnification in every frame, every field of view. So expect to see one here and here and here and here. That's a sign that you've got about this kind of one-to-one, bacterial to soil ratio that everybody's looking for. Often you can have that but not have the protists or the other functional groups present. So it's equally important to pay attention to the protists. So it looks like it could be some possible cyanobacteria or another colony of spores here. It's very important to pay attention to the protist numbers, and a lot of farmers, that are doing small scale sort of market gardening type stuff, protist infusions work really well from them, because they're often the key ingredient that's missing, and they can very rapidly kickstart the nutrient cycling process if that's been lost, because of historic mismanagement or whatever, current management or whatever the reason be. And they're very simple to set up. You do need a bubbler of some kind, something stronger than a fish tank bubbler, but not by much, something like a small pond bubbler. And you can just take something like a 5-gallon bucket. It's the only time I would advise like a flat bottom bucket for this. If you're doing compost teas and stuff, there's a lot more to consider, and you don't want to have surfaces that can the bacteria can easily form biofilms on. Like you really need to avoid that. So for the protozoan infusion, 5-gallon bucket, a load of bubbling stones, or some way to more evenly disperse the air bubbles, and also a bit of a weight to hold the bubbles right at the bottom of the bucket. And then you're going to just go out and you're going to dig around in some leaf litter. Dig around in some leaf litter at night, grab a load of those, just leave them in a bag, but an open bag, so open to ambient temperature, so more or less the same temperature that you collected it from outside. And then in the morning, especially on a really dewy morning, go and harvest, a reasonable amount, no more than just like a decent fistful of grass, cuttings, and green, any green vegetation that you can find that's really dewy. Don't knock the dew off, because a lot of protists, and especially amoebae, hunt in that dew in the early morning. So you can dump in a load of that material into a 5-gallon bucket, turn the bubbler on, bubble away. You definitely need a microscope if you're going to protozoan infusion, and let it bubble away for about 24 hours. Check to see what the population's doing, and the idea is you're gonna see if there's a large number of bacterial colonies going on that have started to establish. Bacteria being, if I just go out of focus slightly, all these things that are moving, all these little dots. So if you observe there to be a high number of bacteria, the protists are sure to start moving out of dormancy, and they're going to start grazing on all these bacteria because they are predators. And check in 24 hours. If there's not very many bacteria, you can add some sort of sweet-like substrate. So it could be molasses, honey would be a good one. Nothing, of course, it can't have any preservatives in it, otherwise you'll start killing things. Any bacterial foods. And then leave it for another 24 hours. And then 48 hours later, check it again. And then you should be hitting that peak of predator-prey interactions where the protist numbers are starting to catch up with, they're starting to become equivalent to the bacterial numbers, and they're starting to overgraze the bacteria. And then at that point, it's perfect time to apply it to your plants, and you can do it on a cool, cold morning, ideally, not freezing, but not in direct sunlight. So ideally early in one of these kind of dewy, like preferentially like spring mornings, I would say. And then yeah, and then hopefully they'll kickstart some of the nutrient cycling again and be a real boon to your plants. So yeah, but I'm gonna end the video here. Like I would say, there's you'll really want to take another look at the soil, like late spring. I would say late spring onwards till like late summer. Is the kind of optimal time to take a look at soil to give an accurate picture of what's going on, because you're interested in these interactions that are happening while your plants are producing, you know, or at their most productive and when they're photosynthesizing at their sort of peak capacity. So yeah, I hope that was informative. Like, thanks. It was nice to meet all the people I did down at the conference. Sorry, I was ill, so we couldn't do the workshop. So I'm happy to do some sort of I'll ask Mary as well. Maybe both of us are up for doing some sort of soil workshop online, because I know quite a lot of you are all living dispersed all around the country. But yeah, maybe we can make up for it in some way. But yeah, all the best and take care of yourselves. Nice to meet you all. All right, bye.